If you're a Unit 3-4 biology student and you've been trying to get your head around CRISPR, you may well be a little bit confused. And you can be forgiven for that because I'm sure, as you found, when you go out on the internet and you look at websites trying to answer those questions that you have about how CRISPR really works, you've found two kinds of websites. Some websites are presented really for the general public. They explain how CRISPR works in a general sort of way, but they don't really answer those nitty-gritty questions. They don't get into enough detail to help you really understand how it really works. And then there are other websites that clearly have been written for molecular biologists, and you read those and you're just met with a wall of terminology that makes them impenetrable. What I hope this video will do is bridge the gap between those two. We're going to go deep. We're going to look at the fine details of CRISPR-Cas9, how it happens naturally in a bacteria as part of their adaptive immune response, and how it can be used in a laboratory by geneticists to edit DNA. We'll look at all of that, and we'll go into more depth than probably you want to go into, but we'll do it slowly. We'll take our time and I'll explain all the terminology as we go along. So sit back and relax and let's get our head around this confusing topic of CRISPR and Cas9. Let's begin by talking about how CRISPR-Cas9 works naturally in a bacteria like Streptococcus pyogenes or E. coli or something like that. Essentially, CRISPR-Cas9 in a bacteria acts as an adaptive immune response. That is, it remembers when a virus has infected the cell in the past, and it keeps a little bit of viral DNA and uses it so that if the same species of virus infects the cell again, it will be able to respond to it much more quickly and more effectively. You can see here in my diagram that over here we have a bacteriophage, that's a kind of virus that infects bacteria, injecting its DNA into this bacterial cell. Like a syringe, it injects its DNA into the bacterial cell. And the first thing that happens is a pair of enzymes called Cas1 and Cas2. They are actually two separate enzymes, but they function together. They're joined at the hip, a bit like thing 1 and thing 2 in the Cat in the Hat. They're always together and they work in concert. And what they will do is they will cut out a region of the virus's DNA called a protospacer and stick it into part of the bacterial chromosome that's called a CRISPR array. Now, we'll talk about the CRISPR array in just a moment, but you'll notice that in the CRISPR array, there are repeats separated by spacers. And so this protospacer will become a spacer in the CRISPR array. The term proto means ahead of or before, and that's exactly what happens. You know, these enzymes Cas1 and Cas2 identify this and think, well, that would be a suitable spacer. Let's turn it into a spacer. So it's a protospacer, and they make it into a spacer. Of course, they don't really think anything. They're enzymes, they don't have brains, but you get the point. Now, the spacer gets inserted at the 5' end of the CRISPR array. All right, that is at the 5' end of the complementary strand of the CRISPR array. So it gets put there, and then they build a new repeat region afterwards. And you'll notice that there's a repeat after each spacer. Spacer, repeat, spacer, repeat, spacer, repeat. Every one of those repeats is exactly the same as all the other repeats. That's why it's called a repeat. And the spacers, of course, are in between them. The term CRISPR array, or CRISPR, the word CRISPR, stands for clustered, regularly interspaced palindromic regions. And let's just break that term down a little bit. Clustered, of course, means all together because this CRISPR array is all in one place on the chromosome. Okay, all these spaces are all together in one cluster. Regularly interspaced is referring to the spaces that are regularly placed between these repeats along the CRISPR region. The palindromic repeats, well, the repeats part of that makes sense, right? Because these repeats are repeated. And the palindromic bit simply means that there are regions within those repeats that read the same on both strands in a 5' to 3' direction. You might remember when you've learned about restriction endonucleases, that restriction endonucleases identify usually a restriction site because it's a palindromic. These repeats are often a site where enzymes can interact. For our understanding of CRISPR-Cas9 here, it doesn't really matter. It's not that important that you understand where and how these things are considered to be palindromic, but that's what the word actually stands for. Now, when it comes to Cas1 and Cas2, taking a protospacer and turning it into a spacer, they don't just cut the DNA randomly in any old place. They cut it at a precise location just upstream of a protospacer adjacent motif. Okay, now a protospacer, this term protospacer is obviously referring to the protospacer. Adjacent means next to, and a motif it's an English word that just means a regularly repeated pattern, like you'd see on wallpaper or something like that. And of course, it's quite common in any DNA to find two guanines together, but the protospacer adjacent motif, at least in Streptococcus pyogenes, is any nucleotide at all followed by guanine. Two guanines together, okay, following after anything, adenine, thymine, cytosine, another guanine, it doesn't really matter. Any nucleotide followed by two guanines, that is the protospacer adjacent motif, or the PAM, as people call it.
Pam? Oh, it's a beautiful name. Pam. Pam. Pam.
Okay, so what happens is these enzymes, Cas1 and Cas2, they'll scan the DNA looking for a PAM site, and when they find it, they'll go upstream, that is, to the 5' end on the complementary strand or the coding strand, and then they'll cut out a section of bases, probably around about 20 to 26 bases long, and then turn that into a spacer. And as I said before, they'll insert the spacer at the 5' end of the CRISPR region and then build a new repeat to the 5' end of that, pushing the spacer further and further toward the 5' end. The CRISPR array then is flexible. If a particular bacteria has been infected by lots of different bacteria, it may have a very long CRISPR array with lots of spacers and repeats. Other bacteria might only have one or a few. I think in some bacteria, people have discovered that they have hundreds of spacers, and in other species of bacteria, there may only be a couple of them. All right, so it's a flexible CRISPR array. From time to time, RNA polymerase will transcribe that CRISPR region, that whole CRISPR array, into an RNA molecule. But we don't call it messenger RNA because it's not going to go to a ribosome and be translated into a polypeptide. We call it pre-CRISPR-RNA. It's a single RNA molecule containing both repeat and spacer regions. And another kind of RNA called what's called unprocessed tracer RNA, which it's been transcribed from another gene somewhere else in the cell. About a quarter of the way around the chromosome, there's another gene that's transcribed by RNA polymerase to make this unprocessed tracer RNA. And the tracer RNA has regions in it that are complementary to regions in the CRISPR RNA, and so it will stick to it. Those complementary regions will stick to the repeat regions in the CRISPR RNA, like I've drawn here. Then an enzyme called RNase comes along and cuts through those repeat regions, giving us a piece of RNA which is made of the RNA from the spacer, the RNA from the repeat, and some of the unprocessed tracer RNA, which we now call tracer RNA. So we have this structure in the cell made really of two pieces of RNA. One piece of RNA, one molecule of RNA, which is spacer RNA and repeat RNA. Okay, that's a single polymer of RNA nucleotides. And then we have another polymer of RNA nucleotides, which is held to it by hydrogen bonds, called tracer RNA. Altogether, we can refer to that as CRISPR:tracer RNA, or CRISPR tracer RNA. Now that molecule gets picked up by an enzyme called Cas9. Cas9 grabs that CRISPR tracer RNA and holds onto it. And now we refer to it as gRNA, or guide RNA. Okay, so guide RNA is CRISPR tracer RNA but it's CRISPR tracer RNA that's attached to a Cas9 enzyme. The Cas9 enzyme itself is made of a single polypeptide chain, but it has 6 different regions that do different things. In particular, we don't need to know what all these regions do, but there's a couple that I think are actually helpful to have a look at. You don't need to remember them, of course, but knowing them and appreciating them is, I think, pretty helpful. Firstly, there's this area here that I've coloured orange that is called the PI, which stands for PAM interacting domain. That's the part of the polypeptide, part of the protein, I should say, which identifies the PAM. Hence the name, PAM interacting. It identifies GG. It has a region in it that has a complementary shape and charges to 2 guanines, and that helps the Cas9 enzyme to identify GG something on a stretch of DNA. These 2 domains here are also really helpful, and by domain we just mean a part of the protein. It sort of it performs a specific function. It is all one polypeptide though, again, just to reiterate that. But HNC and RUVC are what we call nuclease domains. That is, they're the part of the protein that actually cuts the DNA. A lot of the rest of this structure is there just to hold the guide RNA in place in the Cas9 enzyme. And this is what that looks like, or it's really a cartoon representation of what it looks like, because of course, the guide RNA is not all stiff like cardboard, it's all twisted and, and bent up, but it gives you a bit of an image of what it looks like there. You can see there we have both right here, the CRISPR-RNA made of both the RNA that was transcribed from the spacer region of the DNA and the RNA that was transcribed from the repeat region, all as a single polymer of nucleotides, all as a single RNA molecule. And then held to that RNA molecule, we've got a separate polymer of nucleotides here, a separate RNA held to it by complementary base pairing. The way to think about this really is the CRISPR-RNA is kind of like the lenses in my glasses. It's the lenses that do the job of the glasses. It's the reason I have glasses in the first place is because of what the lenses do for me. The purpose of the arms of my glasses is just to hold the lenses to my face. And that's what the tracer RNA does in the guide RNA. The tracer RNA is really there just to anchor the CRISPR RNA to the Cas9 enzyme. It serves as a scaffold, we say, for the CRISPR RNA, holding it to the enzyme. You may have also heard of sgRNA, and there's a bit of confusion around that. A lot of people use the terms sgRNA and gRNA interchangeably, as though they're the same thing. They're not exactly the same thing. And there's even confusion around what SG actually stands for. Some people say that it stands for short guide. Other people say it stands for synthetic guide. And they both actually make sense because it is short and it is synthetic. And by synthetic, I mean it's made in a laboratory. But sgRNA stands for single guide RNA. Now, as I said, it's made in a laboratory. This is what geneticists do when they want to program Cas9 to cut DNA at a specific sequence, which we'll be looking at very soon, actually, is how we use this technology to cut DNA at a specific base sequence where we want the DNA to be cut. But when a geneticist is using Cas9 in this way, rather than using the natural CRISPR tracer RNA that happens naturally in a cell, it's easier just to make a single RNA molecule that will fold up and bend into the shape that they want it to bend into, that mimics CRISPR tracer RNA, but it's made as a single RNA molecule, hence the name single guide RNA. Okay, so if you hear someone saying that it's short guide RNA or synthetic guide RNA, they're wrong. It's single guide RNA. And it effectively, as I said, it does the same thing as guide RNA. In fact, it is a guide RNA. So we could say that single guide RNA is a guide RNA. Not all guide RNAs, though, are single guide RNA. When Cas9 finds some viral DNA, or any DNA for that matter, and it identifies a PAM region, what it will do is it will open the DNA and then see whether the guide RNA that it has is complementary to the sequence of bases upstream of the PAM, but on the opposite strand. Okay, so here's our PAM right here on this strand. It opens the DNA and tries to match its guide RNA to the sequence of nucleotide bases on the other strand, but upstream of the PAM. And if it finds that the sequence is complementary, then Cas9 will cut the DNA on both strands 4 bases upstream of the PAM. Maybe 3 bases or 5 bases, around about 4 bases upstream of the PAM on both strands. Then, of course, that cleaves the DNA, causing what we'd call a double-strand break. A double-strand break, or a DSB, is how you'll see that written in the literature. After that's done, of course, the Cas9 enzyme then leaves the DNA and travels off looking for another PAM site and does the same thing over and over again, constantly looking for PAM sites, opening the DNA, and then seeing whether the guide RNA is complementary to the sequence of bases upstream of the PAM on the opposite strand. Remember that a bacteria uses the CRISPR-Cas9 system as an adaptive immune response. Just like in our immune system, it's really important that our immune system is able to tell the difference between self and non-self. That's just as important for a bacteria. So how does that happen? How can the Cas9 enzyme tell the difference between virus DNA and bacterial DNA? Or to put it another way, if this particular Cas9 enzyme will cut this sequence of nucleotide bases on viral DNA, why won't it cut this sequence of bases on it in its own CRISPR spacer region, right? Because that sequence of bases is identical to that sequence of bases. And they're both DNA, they're both made of the same stuff. So how is Cas9 able to tell the difference? Well, of course, that comes down to the PAM site. Remember, Cas9 won't even interact with the DNA. It won't even open the DNA to see if its guide RNA matches the DNA unless it first finds a PAM site downstream of that region, right? And is looking for that PAM site. When it comes to the spacer region here, it won't even have a look to see if the spacer region is complementary to its guide RNA, because this sequence of bases is not a PAM. It's a different sequence of bases. This is why CRISPR is so ingenious, is because each of these repeats has a sequence that starts with something other than any nucleotide followed by guanine. And that prevents Cas9 from cutting the spacer itself in the CRISPR array. Let's look next at how scientists are able to use CRISPR-Cas9 in order to precisely edit the genome of a species. There are 2 approaches to doing this. We call them non-homologous end joining and homology-directed repair. We'll look at both of those. Remember that time when your parents were out and you stayed home and you played footy in the house and you broke your mum's favourite vase? So you got some super glue and you glued it back together again, hoping that nobody would notice, but you knew that it wasn't really a perfect join. Well, that's pretty much what non-homologous end joining is all about. The non-homologous bit of the name will make more sense once we look at the next way that DNA can be repaired in a cell. But basically it means that you don't need other bits of DNA to act as a template. The end joining, though, is pretty obvious. It just means that the 2 broken bits of DNA can be joined back together end to end. Now, like most things in biotechnology, all the tools needed to do this come naturally from cells. It's the same in bacterial cells and eukaryotic cells. All cells are able to repair breaks in DNA because it happens actually surprisingly commonly. In any typical cell, there will be somewhere between 10 and 50 double-strand breaks in the DNA per day. That's a lot of breaks, and so cells need to be able to fix those breaks, and one way they do that is by non-homologous end joining. Let's have a look at how this works. Now, you don't really need to remember all of the fine details here, I wouldn't imagine, but understanding them and appreciating them, seeing how it works, will give you that depth of understanding to really appreciate how CRISPR is done. All right, so you can see here we've got some DNA that has been broken. It might have been just broken as a double-stranded break, an accidental break, or it may have been cut by a Cas9 enzyme. Either way, what will happen is a protein that exists naturally in the cell called Cu7080 is very strongly attracted to the ends of a piece of DNA. And so Q7080 will stick on those ends, and it, in turn, co-opts, it attracts another protein called DNA protein kinase catalytic subunit, which sticks onto the Q7080. And together, they attract, in turn, a bunch of proteins whose, again, names are weird, but XRCC4 and XLF. And those proteins kind of form, I mean, this is obviously a cartoon version of it, but they form sort of almost like a spring around the two ends of the DNA and join them together. Sort of, you can imagine that happening. They sort of encircle them and bring the two ends together. And then another enzyme called DNA ligase 4 will join those two ends of the DNA. It makes a sugar phosphate bond between the sugar of one nucleotide and the phosphate of another nucleotide, joining the two broken ends back together again. And that sounds really great. And you might be sitting there thinking, well, why would a scientist want to do that? Because it seems kind of pointless. You use Cas9 to cut the DNA, and then the cell is just going to fix it back up again. And that's true. But just like when you broke your mum's vase, the superglue didn't join it back perfectly. And that's what happens here too. Sure, it gets joined back together, but often mistakes are made. Sometimes it's not perfect. Sometimes a few nucleotides are lost. Sometimes an extra nucleotide gets accidentally incorporated into the join. Of course, ligase, it has no knowledge of what it's doing. It's just a protein. It's just making sugar phosphate bonds between bits of DNA. And the process is a little bit sloppy, or error prone, we might say, and it often makes mistakes. As a result of that, a scientist is able to use this technology to hopefully induce a mutation in DNA that might knock out a gene. Now, you've probably heard of knockout genes and wondered what they were. Again, this is a point where there's a lot of confusion about this, because people sort of assume that a knockout, when scientists knock out a gene, that what that means is they're sort of knocking it out of the cell. You know, sort of like knocking the ball out of the park in baseball, right? But it's not that kind of knockout. It's more like a knockout in boxing, where the boxer who's been knocked out is still there in the ring, but they're just unconscious on the floor, right? That's what a knockout gene is like. It's a gene that's been kind of prevented from working. It's a gene that's become non-functional. And there's a couple of ways, of course, that could happen. But one way that I'm sure you're able to understand is just imagine that there was a sequence that went UGC, right? UGC. And you were able to cause a break after the G. Now, it's possible that an extra nucleotide might be added there. So, and if that was an adenine, it would now read UGA, which is a stop codon. And that would make that a knockout gene, or that gene would have been knocked out because we've just created a stop codon that would prevent it from then being transcribed and translated into a protein. We've knocked that gene out. Or a mutation in the promoter region of a gene would probably render it a knocked out gene as well. So there are a number of different ways that this could be used, but it's all based on the idea that while the cell will repair the DNA, it makes a lot of mistakes. And if it makes the right kind of mistake, that lucky accident can sometimes result in disabling a gene or changing a gene in a way that a scientist wants to do it. Another way that cells are able to repair a break in DNA is homology-directed repair. Compared to non-homologous end joining, this is a much less error-prone approach. So it's, ideal for a cell to do this, but cells can't always do homology-directed repair. In order to do it, they need to have a homologous piece of DNA. That is a DNA with the exact same base sequence as the broken piece of DNA. Of course, in our cells, that's usually possible because we've got 2 of each chromosome. We've got a homologous pair of chromosomes. We've always got a backup copy of every length of DNA. In bacteria, it's often not the case because a bacteria has only got 1 circular chromosome, but it is replicated in preparation for binary fission. So at some times of the prokaryotic life cycle, prokaryotic cell cycle, there are 2 copies of each gene. And at that time, homology-directed repair can happen in a bacteria as well. There are 2 processes that can happen in a cell, both under the banner of homology-directed repair. The first of them is called synthesis-dependent strand annealing. It's the simpler of the 2. They're both kind of variations on the same thing. We'll look at both of them, but let's let's have a look at this one first. You can see here, just while we're looking at it, that we've got a broken strand of DNA and we've got another piece of DNA that's homologous to it and has the same sequence of bases on it. I've drawn it upside down. Now, the first thing that happens in homology-directed repair is what we call resection to a Chi site. Now, let's have a look at how that happens. Resection means cutting away. And what happens is the 5' end of both of the broken strands is resectioned by an enzyme called RecBCD. So it'll cut away the 5' end. And you might say, well, why doesn't it keep on going? Why does it stop after a short time? And the reason is that it keeps on resectioning until it hits what's called a Chi site. Now, a Chi site is a length of DNA that has the base sequence GCT, GGT, GG. Okay, GCT, GGT, GG. You can see it here as well. GCT, GGT, GG. So that there is a Chi site, and this here is a Chi site. And this RecBCD enzyme will just cut away the 5' end until it reaches a Chi site, and then it stops. Now, sometimes that would be a long way, right? I've drawn them 5 bases long, but it might be hundreds of bases until it reaches a Chi site. But it will find a Chi site because there are lots of Chi sites. And in fact, somebody studied an E. coli bacteria and found that there were well over 1,000 Chi sites on that one bacterial chromosome. So it may be a long overhang, but it will reach a Chi site at some point. So that's the first step, is resectioning the 5' end back to a Chi site. Now, to illustrate the rest of the process, I'm going to simplify my drawings because my animation skills aren't really quite up to snuff. But I'm sure that you can see that this is exactly the same thing. It's just a bit easier to animate. All right, so with the 5' end resectioned, you can see that now we've got this 3' end here that is sticking out closest to this DNA. Another enzyme called RecA will grab that 3' end and kind of drag it down into the homologous DNA and run along and sort of find where the homologous section is, where the base sequence is complementary. And then an enzyme, DNA polymerase, will do what polymerase enzymes do and extend the 3' end until it reaches a Chi site. And of course, that Chi site is exactly the same as the Chi site that paired this strand back to that point, right? And at that point, the attraction between those two strands is quite strong because there's a lot of complementary bases. And so it gets drawn up and it rejoins the original piece of DNA. So now we have one strand that's been repaired using this homologous strand as a template to do it. But then DNA polymerase also has an exposed 3' end here. And so it can extend that, which it does, extends that until it reaches the other 5' end. And then, of course, DNA ligase will ligate those joins. It will make the sugar phosphate bonds between those joins to give us two now complete pieces of DNA, one that's been repaired using the information in the other. Now remember, we said that scientists can use homology-directed repair to insert a foreign gene into the genome of a species. So how is that done? Well, this is what we do. We're going to create a piece of synthetic DNA. That is, you make it in a laboratory. And because we know exactly where Cas9 is going to cut the DNA, we know the base sequence either side of that, right? And so we make a piece of DNA, or when I say we, they make a piece of DNA that has these two homology arms, that is, stretches of DNA that are homologous to these sections here. But in the middle of it, the piece of DNA that we want to insert into the genome is added between those two homology arms. So what's going to happen if we step our way through this is, again, let's simplify the diagram so we're looking at it. This is exactly the same thing, except it's not a whole extra chromosome. It's just a little fragment that gets inserted into the cell together with the Cas9 enzyme that's got its single guide RNA in it. So the scientist is going to put in a Cas9 enzyme with single guide RNA and these pieces of DNA as well that the cell will use as a template to repair the break created by the Cas9 enzyme. So again, RecBCD is going to resection the 5' end back to a Chi site. And then what will happen is that RecA will drag the 3' end down into that D loop. DNA polymerase will extend the 3' end until it reaches a Chi site. And then at that point, again, mediated by enzymes, that piece of DNA will be broken away from that donor DNA and it'll spontaneously join back to the original DNA because of complementary base pairing there. And then DNA polymerase will extend the 3' end using that repaired piece of DNA as a template to repair the other strand. So we end up with a break repair which has incorporated the foreign gene that was introduced on those synthetic bits of DNA that were added with homology arms. It's pretty cool, isn't it? There's a second process by which cells can repair a break in DNA using homology-directed repair, and this one's called double-strand break repair, or DSBR. I mean, it's a variation on the same theme, but it all happens in one motion. The way that this happens, and again, it happens naturally in cells, is that you can see here that the break in the DNA has been resectioned back to a Chi site by RecBCD, and then what will happen is a D loop forms, and the 3' end and the 5' end both get dragged down into the D loop. DNA polymerase will extend the 3' end until it gets to the Chi site, which is where the 5' end is. So that's the same as what we looked at before. But because the top of the D loop is sitting up against the break in the DNA on the other strand, DNA polymerase can simply extend that 3' end as well using the top of the D loop as a template, and so it can repair both strands really simultaneously. When that happens though, it creates an interesting situation where the DNA has sort of crossed over. It's a little bit like what happens in meiosis when crossing over happens and recombination happens. The same thing happens here. We call this a holiday junction, and a break needs to be made, resulting in a complete piece of fixed DNA and our complete bit of donor DNA, but crossing over has sort of happened, so a little bit of one bit of DNA is on the other one and vice versa. But it doesn't matter, we've still repaired the DNA and in the process incorporated that foreign gene into the DNA. It's pretty fantastic, isn't it? What a fantastic way just to think that cells have this mechanism built right into them so that when those breaks occur, the cell is able to repair the breaks. I mean, that itself, what an amazing concept. There's one more question that a lot of people have about CRISPR-Cas9. I remember when I was learning about CRISPR-Cas9, this really bugged me, right? I understood why Cas9 doesn't cut the bacteria's own DNA because it lacks the PAM site, and it needs that. But why doesn't Cas1, Cas2, why don't they cut the bacteria's own DNA? Why don't they collect a protospacer out of the bacteria? Because all they need is the sequence any nucleotide GG. Surely there's lots of those in the bacteria's own chromosome. Surely. So why don't Cas1 and Cas2 find two guanines together and then cut a protospacer out of that and turn it into a spacer in their CRISPR array? Well, it turns out that the reason is because of those Chi sites. Remember, E. coli, and other species of bacteria too, no doubt, are full, their chromosome is full of Chi sites. There are Chi sites everywhere. Now, of course, the reason for those Chi sites, the main reason for those Chi sites is that so that enzyme RecBCD that resections the 5' end for double-stranded break repair, that's what those Chi sites are there, but they serve another function too, because as it turns out, Cas1 and Cas2 won't make a spacer out of DNA that contains Chi sites. And because there are so many Chi sites in a bacteria's own DNA, that means that Cas1 and Cas2 pretty much don't make spacers out of the bacteria's own DNA. Whereas bacteriophage DNA has little to no Chi sites in it at all, which means that Cas1 and Cas2 will make spacers out of that, but not the bacteria's own DNA. And this is another wonderful example of how this adaptive immune system of a bacteria is able to distinguish self from non-self. Remember, if you're a VCE biology student, you really don't need to remember all the names of the different proteins and the precise names of those different processes and all of that sort of thing. They're just to help you understand it, because you do need to have an understanding of the concept of CRISPR-Cas9 and how it's used as an adaptive immune response in bacteria, and how it's used by biologists to edit the genome of a species. But if this video has deepened your understanding and your appreciation so that you have a better idea of those concepts, then the video served its purpose. Thanks for watching.